Replica plating. Two types of organisms are present, Oxytropes and Prototrophs. In the former group, organisms does not synthesize a particular organic compound required for its growth, whereas in the latter groups are characterized by the ability to synthesize all the compounds needed for their growth. Oxytropes are nutritional mutants, for example, yeast mutant with an inactivated uracil synthesis pathway gene such as orotidine 5-phosphate decarboxylase is a uracil oxytrope. For the growth of such strains, uracil has to be supplemented. In uracil prototroph, such as a wild type strain can grow in the absence of uracil. Beadle and Tatham's Nobel Prize winning work on the one gene one enzyme hypothesis describes the use of oxytrophic genetic markers in molecular genetic studies. If the genetic information of an organism is changed in a stable manner due to mutation, the process is described as mutagenesis. Mutation can result spontaneously in nature. Exposure to mutagens are achieved experimentally using laboratory procedures. The mutagenesis can cause cancer and various other heritable diseases, but it helps in evolution. Hermann Muller, Charlotte Auerbach and J.M. Robson were the pioneers of mutagenesis as a science in the first half of the 20th century. In replica plating technique, colonies were reproduced from master plates choosing any agar based selective medium without losing the special pattern. The technique was developed by Joshua and Esther Lederberg in 1952 for providing the direct evidence for the existence of pre-existing mutations. This technique isolates both nutritional mutants and antibiotic resistant mutants. This is a widely used method in mutation studies to compare the master plate and any secondary plates and to screen for a desired phenotype. The sensitivity of a particular organism to any substance can be monitored by checking the presence of colony on the master and secondary plates. Replica plating is widely used for screening oxytropes and antibiotic resistance. Replica plating allows the observation of microbes under a series of growth conditions. The bacteria are grown in an environment that is not selective for a given mutation. This technique is used to transfer the members of each colony to a selective environment. For the convenience, 30 to 3000 numbers of colonies were replica plated due to the difficulty in streaking each out individually onto a separate plate. Method of replica plating. A simple velveteen covered colony transfer device is used to transfer the colonies in nutrient agar medium supplemented with or without a particular antibiotic or nutrient. The fibers of velvet act as fine inoculating needles picking up the bacterial cells from the surface of this master plate. The velvet with its attached microbes is then touched to the surface of a sterile agar plate, inoculating it. In this manner, microbes can be repeatedly stamped onto media of differing composition. By comparing the presence of colonies following incubation, we can indirectly determine the mutant colonies by their absence in the selective environment. A colony that develops on a complete medium fails to develop on a minimal medium that lacks a specific growth factor. The occurrence of a nutritional mutant is indicated. The microbes that do not grow on the minimal medium represent oxytrophic strains. This method has been applied in numerous experiments to identify the occurrence of mutations. Many of the biochemical pathways in microbes were elucidated in this way by using nutritional mutants.